Welcome back. Today we're digging into a fascinating study about fats, T cells, and a kind of cell death called ferroptosis, and how the balance of different dietary fats can change how well our immune system works.
That's right. We usually hear that polyunsaturated fats are good and monounsaturated fats are good too. Fish, nuts, olive oil, avocados. But this paper suggests the story gets more complicated when it comes to immunity. It's not just whether a fat is healthy for the heart. It's about the ratio of polyunsaturated to monounsaturated fats and how that affects T cell survival and immune responses.
Let's unpack ferroptosis first. It's a form of programmed cell death, but very different from the classic apoptosis most people have heard of. Ferroptosis is iron-dependent and driven by oxidation of certain fatty acids in cell membranes. Picture the cell membrane as a chain-link fence. Polyunsaturated fats, PUFAs, have multiple double bonds, which makes them like weak spots along that fence. Iron chemistry can set off a chain reaction oxidizing those double bonds, creating lipid hydroperoxides that eventually poke holes in the membrane.
That's a great image. And the cell has a firefighter of sorts, an enzyme called GPX4. GPX4 neutralizes those lipid hydroperoxides before they become lethal. So the balance between how many PUFA weak spots you have in the membrane and the cell's ability to neutralize oxidative damage determines whether a T cell falls to ferroptosis.
The researchers noticed something almost by accident. They were comparing T cells from mice that had been fed different standard lab diets that are considered nutritionally complete and often used interchangeably. But T cells from those mice showed dramatically different sensitivity to ferroptosis.
Wait, that's interesting. So diets we assume are equivalent in experiments actually change T cell vulnerability?
Exactly. The team put young mice on 3 common rodent diets, SF-NAI31, SF00-100, and SF-AIN93G, which, crucially, differ in their PUFA-MUFA ratios, high, medium, and low, respectively. The mice looked the same by most measures, but when the researchers challenged splenic T cells ex vivo with increasing amounts of a GPX4 inhibitor, the cell susceptibility diverged. They defined a ferroptosis resistance index, the inhibitor concentration needed to kill half the cells.
And the result?
T cells from mice on the high PUFA-MUFA diet were 2 to 3-fold more sensitive, lower resistance indexes, than those on the low-ratio diet. They confirmed it with another ferroptosis inducer and showed that a ferroptosis inhibitor could rescue the cells, so it was definitely ferroptosis. Interestingly, apoptosis responses were the same across diets, so this was specific to the ferroptotic pathway.
So diet shifted the balance between PUFAs and MUFAs in T cells, changing their susceptibility to oxidative membrane damage. Did they test this in living animals?
They did. Mice with T cell-specific deletion of GPX4, meaning their T cells can't neutralize lipid hydroperoxides, showed dramatic diet-dependent T cell loss. On the low PUFA-MUFA diet, those mice retained about 35% of their CD8 plus T cells relative to controls, 3.5 times more than mice on the high-ratio diet. CD4 plus cells followed a similar but milder pattern.
That's a big difference. But does it translate to real immune function? Numbers are one thing, but what about the immune response?
The authors tested that using TFH cells, follicular helper T cells that help B cells make high-quality antibodies and are known to be especially vulnerable to ferroptosis. They transferred GPX4 deficient T cells into recipient mice that couldn't make their own TFH cells. On the low-ratio diet, the transferred cells produced almost normal levels of TFH cells, several times higher than on the high-ratio diet. The team ruled out other explanations like gene expression or microbiota differences and showed that ferroptosis resistance tracked inversely with the dietary PUFA-MUFA ratio.
How did the dietary fats actually get into the T cells?
They measured fatty acid composition in the T cell zones of lymphoid organs and found the PUFA-MUFA ratio there mirrored the diet. Free fatty acids in those zones correlated with corresponding phospholipids. The picture is that dietary PUFAs arrive in lymphoid tissue, get incorporated into T cell membrane phospholipids, and create more substrate for lipid peroxidation.
That mechanism makes sense. Any implications for cancer immunity?
Yes, and this is another surprising insight. In mice with melanoma that elicit adaptive immune responses, tumors grew significantly slower in mice on the low PUFA-MUFA diet. Those mice had more effector CD8 plus T cells and much greater clonal expansion. They also did experiments using human CAR T cells in immunodeficient mice engrafted with human leukemia. CAR T cells in low-ratio fed mice had lower lipid ROS and 2 to 4-fold better persistence in circulation. With CAR T therapy, low-ratio fed mice showed markedly better tumor control and survival. Without CAR T therapy, the leukemia progressed equally regardless of diet.
So the diet didn't change the tumor directly, it changed how effective T cells were against the tumor.
Exactly. And they even looked in people. In 24 healthy young adults, plasma PUFA-MUFA ratios in phospholipids correlated positively with T cell lipid ROS and negatively with ferroptosis resistance. A cohort of 12 children showed the same pattern.
Notably, BMI didn't predict ferroptosis resistance in either group. It was the lipid composition that mattered.
That's a striking takeaway. It's the types of fats in circulation, not body weight, that correlate with T-cell vulnerability.
The lead researcher, Professor Du, summed it up. The kinds of fats you eat change the fat composition inside your T cells, and those changes can make T cells either weaker or stronger in terms of immune protection. She suggested dietary changes could potentially boost vaccines and cancer therapies.
That's provocative. So while PUFAs and MUFAs are both healthy in many ways, when it comes to immunity and ferroptosis, their balance matters a lot. And even standard lab diets can shift experimental outcomes.
Right. The study not only reveals a cellular mechanism, but also highlights something practical for research and therapy. Small changes in dietary lipid composition can have outsized effects on T-cell viability and function.
Final thought?
If you're thinking about immunity, don't just ask how much fat, ask what kind. The PUFA-MUFA balance can tilt T cells toward survival or iron-driven death, with real consequences for antibodies, tumor control, and immunotherapies.
That's a powerful reminder that diet and immunity are intertwined in ways we're still uncovering. Thanks for listening. We'll keep following this line of work as it develops.